Good morning, everyone. My name is Roshan Ramoutar. My team members and I each hold a BSc in physics, and under the keen guidance of Dr. Davinder Sharma of the physics department, we conducted a study on an ongoing research on microbial fuel cells. Being a relatively young technology, not many people are aware of its existence. So I'll introduce the concept of the MFC. As its name suggests, it is a fuel cell, so it converts chemical energy into electrical energy, but it does so by the work of microbes. An MFC reactor takes advantage of the fact that when microbes respire anaerobically, they convert glucose and water into carbon dioxide, hydrogen protons, and free electrons. In our study, we utilized the Dual Chamber MFC configuration, where wastewater containing naturally occurring microbes is placed in one chamber with an anode and sealed off from the atmosphere. The second chamber contained distilled water with the cathode and left open to the atmosphere. A special membrane, known as a proton exchange membrane, or PEM, completely separates the contents of both chambers. This membrane allows only hydrogen atoms to pass through. When microbes metabolize wastewater, the free electrons are captured by the anode, and hydrogen protons diffuse across the PEM into the cathode chamber. Within the cathode chamber, the hydrogen protons combine with oxygen protons and free electrons to produce pure H2O. The free electrons come from the anode chamber across the circuit that we created, and the affinity for electrons by the cathode chamber gives rise to a potential difference between both chambers, producing the electrical current. The objective of our study was to enhance the performance of the dual chamber MFC for electricity generation by varying three parameters: electrode material, electrode shape and size, and wastewater origin. To do this, we used glass chambers of 150 milliliter capacity and two electrode materials, aluminum mesh and platinum carbon 40%. For the aluminum mesh, we used anode to cathode area ratios of 2 to 1 and 6 to 1, while for platinum carbon electrodes, we used a ratio of only 2 to 1. In addition to material, we varied the shape of the electrodes using a flat, square, cylindrical, and coiled arrangement. A material known as Nafion 212 was used for the PEM, and a small fish tank pump was used to constantly oxygenate the distilled water in the cathode chamber. Wastewater samples were taken from various locations around Trinidad: Cascadoux trees, Nariva swamp, Caroni swamp, Guaracara River, and a cow farm located in Cunupia. Here is a view of an assembled reactor with the anode chamber with a square flat aluminum electrode sealed off from the atmosphere, separated from the cathode chamber by the PEM sheet, and all held together by a clamp. Here we see an MFC reactor in operation with a voltmeter between the two electrodes with an actual reading from our setup. Preliminary results show that the cylindrical shape for the aluminum electrodes were far more efficient at producing a potential difference, so this shape was maintained for the rest of the study. After setting up of components, we calibrated it by using distilled water in both chambers. We then moved to one wastewater sample in each of the electrode configurations. We recorded the potential difference between the chambers every hour, every day, until the voltage diminished. This took about 3 to 5 days depending on the sample. The samples were chosen based on their backgrounds and type of waste they may be exposed to. Nariva swamp and Cascadeaux trees were chosen for their seclusion and remoteness to human interference. It is expected to be populated by microbes that feed off natural waste. The Guaracara River is located amongst industrial sites where artificial waste from civilian and industry may run off into the river. The cow farm in Cunupia should contain feces, urine, and various pesticides, and therefore should be home to microbes that live off these compounds. Now let us recognize the workforce, the microbes. Based on previous studies done by Bruce E. Logan in 2004, smaller reactors are more efficient than larger ones, since it kept the environment inside the chamber constant for longer. The targeted microbes are those of the benthic zone. This is a layer below the surface of waterways where oxygen and light is scarce, so the microbes that live here are already used to oxygen deficient environment. To access this layer, we use a spade to dig about 1 foot deep to remove samples, place them into a sealable container isolated from oxygen and light. After testing our samples and ratio combinations, we selected the more attractive results. Here we see Nariva swamp reaching a high of about 350 millivolts initially, then falling off gently for the next 40 hours, while Cascadeaux trace produced a relatively steady voltage of 220 millivolts throughout the study. For Guaracara River, we see the aluminum mesh 2:1 and 6:1 fluctuating greatly for 60 hours. Their voltages were higher than that of the platinum carbon, but were unstable. Voltages for the platinum carbon raised gently and plateaued at around 580 millivolts for the next 40 hours before falling off. Caroni sample shows a similar trend where aluminum mesh 2:1 achieved higher voltages than platinum carbon, while platinum carbon was more stable. Aluminum mesh 6:1 maintained a low voltage throughout. Results from the cow farm sample also followed a similar trend, except that in all cases voltages were much higher. We see the aluminum mesh 6:1 peaking at various times, reaching about 870 millivolts, while the aluminum mesh 2:1 maintained a voltage of about 550 millivolts. The platinum carbon was the most stable at about 800 millivolts. At this point, we decided to toss in an oddball by running an aluminum mesh ratio of 10:1, just to observe its behavior. We see the potential difference rise sharply for the first 15 hours to almost 1000 millivolts and stayed there for the next 20 hours before dropping off drastically and repeating the pattern. We gather from the results that a larger anode to cathode ratio can generate a larger voltage, but not for long periods, while smaller ratios saw less fluctuations. The material plays a role in the stability where platinum carbon electrodes maintain more stable voltages. From the graphs, we can see that animal waste is the most efficient power source in our study. There are many possible applications of MFC technology in the Caribbean, and further research could see an upscaled version or even a combination of smaller reactors to provide a solution to waste management and power generation simultaneously. In the Caribbean, there are many small-scale farms located far from cities and towns and may even be off-grid. Acquiring power is a challenge. The MFC could help provide these needs at low cost, with the only fuel being waste. Farmers' standards of living could improve using organic means of power generation. In conclusion, the most attractive results came from the cow farm sample, suggesting these microbes are the most efficient as a fuel source for the MFC. The highest voltage acquired was from the aluminum mesh 6:1 ratio of 950 millivolts, but was very unstable, while the platinum carbon electrodes maintained a more stable and possibly useful voltage throughout the study. So this brings me to the end of my presentation. I thank you for your attention. Any questions?
Thank you very much, Roshan, for that very informative presentation. Any questions? I did not see any from the chat menu.
Well, I have a question. I'm Durjari Prasad Chakravarti, one of the moderators. What is your idea about the scale up? You said that smaller reactor is working much more better way than a bigger one. So yeah, I just want to get clarified.
So why? Are you asking why?
What is your idea about the scale up?
Oh, right. Scaling up could be a problem because the electrode needs to come into contact, or at least close proximity to the molecules in the chamber. So they need to come into contact with microbes feeding off the wastewater. So scaling up would mean you would have to use a more complex electrode in the sample. But according to previous research, when using larger chambers, the reactions are not as efficient. So either they are slowed down by distance that they have to travel, or pH levels, or availability of glucose for them to feed off. So there are many factors that will contribute to the smaller chambers being more power efficient.
All right. Okay, thank you very much.
I can allow one more question if any of the attendees have any question. One more. If no more questions, you can also email your questions to Roshan. Thank you very much, Roshan.
No problem, I'm available. You can always email any concerns or questions.
Right, and thank you for keeping on time your presentation.